Okay, so I'm in a hotel room in North Carolina, so forgive me if this isn't the best video ever. But I got some paper, and I made us a little hypothetical situation. Hopefully this will help explain it to you. So, in my example, you've got this enzyme called shapase, and you have these two proteins. You have circle and diamond protein. And when circle protein is exposed to UV light, it is converted into diamond protein. And hopefully my hands aren't shaking too much and you can actually read this. And so in a healthy cell, the ideal is that these two proteins will be equal in their concentration in the cell. So the idea here is that shapase converts the diamond cell, or the diamond protein rather, to the circle protein. So what you would have is your perfect situation here is a healthy cell, right? You have your equal components of circle protein and diamond protein. And when the cell is exposed to UV light, then you have a greater concentration of diamond protein than you do have circle protein. So here's our example of an unhealthy cell. So the question is, okay, well now we have an excess of diamond protein. How do we get back to the healthy cell standpoint where we have equal parts of both proteins? So shapase, like I said before, is going to be what converts your diamond protein back into your circle protein to get us there. So what happens if there's no enzyme regulation whatsoever? Then you're going to end up with just all circle proteins, right? Because if shapase is just constantly converting diamond proteins into circle proteins, and you have no way to regulate it, you're going to end up with all circle proteins, and that's not a healthy cell, because a healthy cell has to have equal parts of both protein. So there are a couple ways that you talk about in your specific Facebook question as to how that happens. So the first is allosteric, and what that means is that you're going to have a change in the shape, usually conformational change, of the enzyme by something. So in this case, it's going to be a direct inhibition effect, where a circle protein will directly inhibit the effect of our enzyme shapase. And so it binds to shapase at, and I have it here, wrong finger, I have it here at a non-competitive site, right? So when it's bound, diamond protein no longer converts to circle protein. So that means that when you're at this point where you're having an excess of circle protein, it will turn off, due to the high concentration of circle protein, will turn off shapase so that it's no longer converting it. And that will ideally prevent you from going past this level where there's equal amounts. So the goal is to have a healthy cell by stopping too much conversion from happening. And so, like I said, that often happens as a conformational change, so it'll change the shape of the active site, and so you see it's a square now, a large square, because I guess a diamond could technically fit into a square spot. But the point is that it doesn't go there anymore, it no longer binds, it's too weird of a conformation, it contorts in some way, whatever. Allosteric inhibition means that it can no longer bind to that site. And I heard a Facebook ding go off, so I'm hoping that someone else didn't just... Okay, it's just you telling me that you want me to answer your question. Okay, good. I'm glad I'm making this video then. So the second way is covalent modification. And in cells, this is overwhelmingly done by ATP. So let's say in our hypothetical example that this reaction requires energy of some form. So covalent modification could be done in one of two ways. You could have ATP binding site or an ADP binding site, because they would both be forms of a molecule covalently binding to your enzyme in order to either upregulate or downregulate its activity. So let's look first at the ATP binding site. So the example with an ATP binding site, you've got a little spot for it right here on our shapase, and when ATP binds here, and then you have your what did I say? I'm sorry. Your diamond protein, right, would bind, and you've got your ATP, and because the reaction requires energy, there you go. It would produce your circle protein to get you to that healthy cell state. But if there's no ATP bound, the reaction requires energy, so nothing's going to happen, even if your diamond protein is bound. So that means that the reaction is necessarily regulated by the presence or absence of ATP. And then the converse example would be that ADP would bind, and when there's not enough energy in the cell for the reaction to proceed, because usually it'd be something like you've used up the ATP on your reaction, so the cell says, okay, if I'm using this much ATP, it must be because this reaction's happening a lot. We want to slow it down so that it doesn't, again, get to this state where you have all circle protein. And so we have this ADP binding site, and as you can see from this particular example, when the ADP is bound to the enzyme, it kind of sterically hinders the binding site, and so the diamond protein can't bind there, the ATP won't bind to its binding site if it were to have both, or it could just have the one where just the lack of energy sterically inhibits the binding of your substrate protein. It could go either way. It doesn't have to have both kinds of binding sites. Sorry if that was a little unclear. It could be just one or the other, or it could have both. And so yeah, then that would prohibit the reaction from proceeding. And the third way, I do with this piece of paper, third way is the variation in the amount synthesized. So the enzyme obviously has to come from somewhere, and it comes from a parent DNA strand, which gets converted to your mRNA, and then into your shape-base protein. So what would usually happen, it would usually be through another enzyme, which I know kind of makes things a little confusing, but we have this random little enzyme I drew here, and depending on which substrate is present in a higher concentration, it will either upregulate it or downregulate it. So the presence of the diamond-shaped protein is going to upregulate it, obviously, because increased presence of diamond-shaped protein will mean that you would want more activity to transfer those into circle proteins, right, to get, again, healthy cell state. I'm hoping this isn't too confusing. I'm sorry, I totally made this enzyme up on the fly, and it might be really random and confusing. I'm really sorry about that. Or if there's circle protein, it would downregulate, and because you wouldn't want to end up with all circle proteins. And what's so genius about most cells is that they usually will have both of these things, because in what this enzyme will do is then it will bind to the DNA, and so if there is a circle protein bound to it, it'll go bind to the DNA, and then once a triangle protein binds to it, it will unbind from the DNA. And whether or not it's bound will influence whether or not polymerase is able to bind to the DNA strand to actually make mRNA out of it, so that it can make shape-base. So the concentration of shape-base in the molecule is directly influenced by what the concentration of the substrate is. And so the reason why most cells have both is because if it's 50-50 concentration, then that means you should have equal amounts of this bound and unbound to the DNA. So it will keep shape-base just kind of at a constant concentration, maintaining equilibrium, rather than kicking into overdrive in the case where there is too much diamond protein because of UV regulation, or it will be not being produced as much in the case of too much circle protein because it's been working too hard. Now we've gone past the point of equilibrium, back to being an unhealthy cell, just on the wrong side, not because of UV anymore, but because the enzyme did too much work. So I hope that makes sense, and feel free to ask me questions and whatnot, and yeah, I hope that helps. Let me know.